morning, everybody. Before I start, I want to show you two pictures. Here it is. Can anyone tell me the difference between these two pictures?
The height.
Yes, maybe. Is there any difference?
The height of the tree.
It's quite close to.
The shape.
Yes, it's right. It's the shape, and you can see that from the left tree is a normal tree which is growing strictly upwards, while the right one is a bent tree. Why are they like this? It is probably related to the supporting material inside the tree. And one more question. Can anyone tell me what is the major component for support in trees?
Silent.
Silent, yes. And here shows a cross-section of a tree trunk. And yes, you told us that silent is the major supporting material in tree trunks. And in fact, the major component inside silent is in fact lignin. And lignin play an important role in higher plants for support, transport of water and solutes, providing physical barrier against pathogen, as well as making a significant contribution to the carbon input in soil. And however, lignin is an undesirable wood content in formation of paper, as it is quite resistant to decay by microorganisms. With the help of modern technology, we can the lignin content, the composition, as well as the conformation can be altered through genetic engineering. And in addition to this, the subunit of lignin can also be changed through downregulation of related enzymes. And that's why the effect of altered lignin on the major ecological process of decomposition is an obvious target for investigation. There were several experiments done in the past to study the effect of genetic modification to lignin biosynthesis and the activity of decomposer organisms to provide information on understanding of ecological effects. One experiment was done in 2001. Triplet of four lines of tobacco plants were studied, in which 3 of them were introduced with one of 3 crucial enzymes. They were reduced CAD, reduced COMT, and reduced CCR. And these plant materials were included in different soil under laboratory conditions for 77 days. Another experiment was done 1 year later to study a 4-year-long field trials performance of pulp trees with genetic modification to lignin biosynthesis. And this time the anti-sense gene of enzyme, CAD and COMT, were involved. And 3 years later, a researcher performed another experiment to study, to test whether changes in the conformation of lignin that reduced protection from enzymatic attack is afforded to more labile components like polysaccharides. And here is the results of the 3 experiments. The result shows that the decomposition rates of modified plants decompose more rapidly than the unmodified plants. And among the 3 modified plants with reduced CCR has the fastest rate of decomposition. And here is the data showing that the decomposition rates in different soil of stem section from the tobacco plants. You can see that the order is following with reduced CCR is the highest rate, followed by reduced COMT, and then reduced CAD, and finally the wild type, which is the unmodified part. And however, there's an exception in soil-free. You can see that the overall performance of decomposition rate is relatively slower in soil-free because of its acidity. Soil-free has the highest acidity, and as plant material was added to the soil as a relatively large amount in a small much bed, so the area of contact between the plant material and the soil will become relatively smaller, and so any physical changes in the soil condition may make a significant contribution to the decomposition rate. Another notable changes in chemical composition can be revealed by the solid-state carbon-13 nuclear magnetic resonance spectroscopy of stem section is shown here. You can see that there is an obvious difference shown by the green arrow. The absorbance is quite different for the reduced CCR compared to the other types. And the research still remember the decomposition order I've just mentioned. The researcher tried to relate the change in the chemical composition in the reduced CCR to the decomposition rate. They conclude that the alteration of the lignin composition in reduced CCR is due to the change in composition of the reduced CCR. And to conclude, researchers try to relate their experimental results to fit their hypothesis. However, even though a lot of process of lignin biosynthesis has been studied these days, however, none of them can provide detailed understanding of the process of decomposition and lignin biosynthesis. So I suggest long time and more data field-based investigation are needed. And that's all of my presentation. And here